Is this thing on? In 1902, Austrian botanist Gottlieb Haberlange proposed a theory that every plant cell contained all the genetic material necessary to regenerate and give rise to a new complete organism. This theory, known as totipotency, caused Haberlange to become known as the father of plant cell culture. By his death in 1945, his theories were already well on their way to being proven. What he couldn't have predicted, however, is that in the over 81 years since his death, his discoveries would be used to rewrite the very genes that make up plants, giving rise to the industry of genetically modified crops. In 1939, at the Rockefeller Institute for Medical Research in New Jersey, Philip White developed a method to create a plant tissue culture with potential for unlimited development. Remarkably, within these cultures, roots and shoots began to develop. It was at this point that researchers realized that totipotency was possible and set in motion the wheels of the first successful plant regeneration. In 1959, Folke Sko and Carlos O. Miller, when studying plant hormones, discovered that the ratio of hormones actin and kinetin in a culture could affect the development of roots and shoots. With this discovery, researchers were well on their way to generating complete plants from tissue cultures. In 1965, the University of Wisconsin, Wimla Vassel and Albert Hildebrand successfully regenerated a full tobacco plant from cultured cells. In 1970 in Berlin, Guillaume Box-Husseman and Jakob Reiner fully proved Haberlange's prediction of totipotency by developing single plant cells into embryos. Meanwhile, research on the bacterium Agrobacterium tumefaciens was on the way to proving that gene transfer was possible. In 1977, Mary Del Chilton, Milton Gordon, and Eugene Nestor discovered that Agrobacterium transfers DNA directly into plant genomes. This discovery allowed Agrobacterium to be used as a tool for genetic transfer. In 1983, groups led by Chilton, Jeff Shell, and Rob Horsch successfully created the first engineered genes that functioned inside plant cells. By 1990, virtually any crop could be genetically engineered and subsequently cloned as many times as necessary, giving rise to the modern GMO industry.
I'm here in a school cafeteria where tons of GMOs are served to children every single day. Throughout the US, it's a very controversial topic. Some people think that they're bad for you. Some people think they're good for you. And there's not a whole lot of evidence to support either side completely. But unlike a lot of modern biotechnology, GMOs are widely used all across the US and all across the world and are not receiving a whole lot of legislative backlash.
So what exactly are GMO crops? How are they made? And why do they matter? GMO is any organism whose DNA has been changed using modern forms of biotechnology. A GM crop is a type of GMO that is used in the form of a plant engineered to express a specific new trait, like insect resistance or herbicide tolerance. These crops were developed to make agriculture more efficient, which ultimately help farmers protect plants from insects, reduce losses, and produce more food. So even though the science behind GMOs is very complex, the main process behind it can be broken down into 6 primary steps. The first step is isolating the target gene, where scientists find a useful gene, like the Bt gene from Bacillus thuringiensis, which has the ability to kill certain insects. Now they identify the organism that has the natural trait, locate the exact gene in its DNA, and design a version that works well in plants. The second step is building the DNA package. During this step, a promoter turns the gene on, and a terminator is present to end transcription. And there's also a selectable marker so researchers can identify which cells took up the new DNA. The third step is inserting this DNA into the plant cells. This can be done using Agrobacterium, a gene gun, or new CRISPR technology that has been developed in the last 10 to 15 years. But all 3 methods allow this process and step to take place. The fourth step is growing whole plants from these cells. This is where the transformed cells are grown in tissue culture, and only the cells that receive the new gene survive. And they eventually regenerate into full plants carrying the trait. The fifth step is testing the GM plants themselves, which scientists confirm the gene is stable, they measure the protein levels, and test the trait. And then finally, this process and GM crops need to be approved and regulated in order for the crop to be released to the public. So before reaching the farms, they go through safety, environmental, and field testing and must be approved by worldwide regulatory agencies. So one of the best examples of how GM crops work is through Bt crops, as I previously mentioned. These plants produce Bt proteins that bind to receptors in certain insects' guts, which kill the insect, but leave the humans and other organisms completely unharmed. GM crops like soybean, corn, cotton, and canola are used worldwide, They show up in everyday foods like corn syrup, cornstarch, soybean oil, canola oil, and even in the textile industry through cotton. Engineers also develop GM crops to increase food production, reduce pest damage, and lower chemical use to improve nutrition. For the average person, GM crops can mean more affordable food, improved nutritional options, as well as reducing pesticide use. But there are also concerns like the evolution of resistant pests and economic impacts from the regulations. But overall, GM crops play a major role in modern agriculture and continue to grow in cultural and worldwide importance.
There are many pros and cons to the use of GMO crops. For example, some pros that are being exhibited today are that corn is currently being genetically engineered in Africa to be able to survive and thrive in areas with very low amounts of growable food, in which there is a lot of starvation. So overall, I think we can all agree that that is something very beneficial. But a lot of people snap back with that, saying that something like that can become invasive to that area because that species is designed to be able to dominate an area that already doesn't have a lot of organisms. And this could pose serious threats to environments. So with me right here, I have Kier. Say hi.
Hello.
So how much pounds of genetically modified food do you think you digest a year as an American? Like pounds of food.
Pounds? Maybe like 300.
300? That's pretty close. It's 193 pounds per year. That's a good guess. I think a lot of people would guess a lot lower than that. And I have one more question for you. Do you think genetically modified organisms and crops are good for you or bad for you?
Some could be good, some could be bad.
That's a valid answer. It's a good answer. Very well educated. Thank you, man. 48% of Americans think that GMOs are okay, but 45% think that they are just completely risky and they're not really worth it. This shows the controversiality of GMOs, bringing me to its biggest hurdle that it's going to need to face, is its public acceptance. There have even been talks of legislation that would ban GMOs entirely, and that would be detrimental to the technology. The topic of genetically modified organisms connects directly to every single unit of AP biology. First of all, cellular genetics, of course. Unit 3, genetics is the basis of GMO technology. GMO stands for genetically modified organism. The idea of a GMO is that humans synthetically alter an organism's genetics in order for a specific outcome. Secondly, gene expression and regulation, Unit 6, is affected by genetic modifications, as they change genes that are then expressed, some of which may be regulatory genes that code for inhibitor or activator proteins. This brings us to Unit 1, chemistry of life, because these proteins discussed are made of amino acid monomers that form together to make a polypeptide or a protein, as discussed in the chemistry of life topic. Due to these changes in protein material that genetic modifications have, a change in function is subsequently caused, as seen in Unit 2, cell structure and function, where structure's impact in function is heavily emphasized. This change in function can impact cell communication and the cell cycle, Unit 4, by changing proteins involved in regulation, response, and secretion of ligands. In the grand scheme, genetically modified organisms with the cellular chart changes mentioned above will likely have higher evolutionary fitness than the wild-type version of the plants because they are designed to grow more easy and be more resilient and just have more cells in general. As you can see, this cucumber or zucchini, we can't tell what it is. It's very, very big. If you look at natural zucchini or natural cucumbers, they are nowhere near as big. And because of that, these would likely have higher evolutionary fitness due to their modifications to be more resistant to bugs, pesticides, and harsh conditions, giving them an edge in natural selection. Unit 7, kept domesticated, these organisms that are more fit than others can become invasive to a region and mess up the ecology of that area. Ecology, Unit 8. This reason why organisms are harmful because they can reproduce, causing the next generation to inherit the same genes that gave the previous generation their unfit fitness. An idea elaborated on in Unit 5, heredity, which is a unit that elaborates on how genes are passed from generation to generation. Just like if this plant, before it was picked, reproduced with another plant, the genetically modified genes passed down to the offspring. GM crops have been a consistently heated topic in almost all of mainstream media. And there have been studies on rats, not humans, that have showed possible correlation to hepatic, pancreatic, renal, or reproductive effects, and possibly even an increase in cancer risk. But even the researchers who researched that study said that multiple generation, longer form tests must be done to make conclusive conclusions. It is your responsibility to figure out what you believe on whether they are healthy or not, and make your decisions accordingly. Thanks for watching. It's a cucumber.